How do Neurotech sensors measure local field potential? Imagine being able to listen in on the brain's conversations, not by eavesdropping on individual neurons but by tuning into the collective chatter of many. That's what neurotechnology sensors do when they measure local field potentials. These sensors are designed to pick up the electrical signals generated by groups of neurons working together in a specific brain area. Instead of focusing on single neuron spikes, they listen to the overall electrical currents flowing in the space outside the neurons, which mainly reflected synaptic activity and slower changes in the membrane potential. The process starts with tiny microelectrodes, often just a few micrometers in diameter, that are carefully inserted into the brain tissue. These electrodes are built to detect voltage changes in the fluid surrounding neurons. When neurons are active, they produce electrical signals that cause tiny voltage fluctuations in the extracellular space. The electrodes pick up these signals, which are broadband, meaning they include a wide range of frequencies from about 1 Hz up to several thousand Hz. The raw signals captured are very complex, so they are filtered to focus on the low-frequency components, usually between 1 and 50 Hz, which are characteristic of local field potentials. This filtering helps separate the signals from the faster spikes of individual neurons, which are typically in the higher frequency range. Once filtered, the signals reveal the collective activity of neuron groups, showing how different parts of the brain communicate and coordinate. These signals can be analyzed to identify patterns like oscillations, bursts, or changes in power within specific frequency bands. Such features tell us about the balance of excitation and inhibition in neural circuits, giving clues about how the brain processes information. Neurotech sensors used for measuring local field potentials often come in arrays, with multiple microelectrodes arranged in a grid or linear pattern. This setup allows scientists to record from several sites at once, creating a map of activity across different brain regions. These sensors need to be highly sensitive and stable because the voltage changes they detect are very small, often just a few microvolts, and they must do so with millisecond precision to capture rapid brain dynamics. In real-world applications, measuring local field potentials helps researchers understand brain function in both healthy and diseased states. For example, in brain-machine interfaces, these signals are decoded to control prosthetic limbs or computer cursors. In clinical settings, they assist in monitoring brain activity during surgery or evaluating neural responses to drugs. Although recording these signals requires inserting electrodes into the brain, which is invasive, it provides detailed information that is otherwise impossible to obtain non-invasively. To sum up, Neurotech sensors measure local field potentials by detecting the tiny voltage fluctuations in the extracellular space caused by the synchronized activity of neuron groups. They filter and analyze these signals to reveal how different parts of the brain work together, providing a window into the brain's local processing with high temporal and spatial detail.